Okay, well, thank you, Jill, for really causing us to really think about this. I'm not going to really talk about smoking per se during my talk. What I'm going to do is try to lay out essentially a framework on how to organize your thinking today and what is going to be the important pieces of evidence that you're going to use that you want to present to the decision makers, present to the public, that shows that there is truly a heritable risk problem, and then try to figure out if there are policy initiatives or anything that we can go forward with to actually take some action or try to reduce the amount of exposure, if you will. So we'll talk about susceptible populations today. We'll talk about the different types of smoking, the different products and things like that. So keep that in mind while I go through this. This is going to be the way we're going to sort of organize our talking at the end of the day. To really understand the framework that I'm talking about, you have to understand what we're going to talk about is really the elements of risk assessment. So what you want to do is you're going to try to go through a process, which we're going to show you in a minute, the framework, for actually estimating the probability of exposure to a hazard and the resulting public health impact. So what we're trying to do is actually focus on risk. We all know tobacco smoking and the related products are bad. What we've got to figure out is, what can we do about reducing the amount of smoking and the use of those products? Because society has essentially said, for adults anyway, smoking is not going to be banned. So we've got to figure out a way that we make them very aware that there is a problem, not only for themselves, but for future generations. And I think that's really what EMGS is all about, is trying to figure out what's happening down the line. I just want to give a quick overview of risk analysis. I'm not going to dwell on this very long, but essentially there are several phases you go through. There's a planning scoping phase, you go through a problem formulation phase into the risk assessment itself. That's actually talking about the actual scientific problems you're going to talk about. And these are the four main steps that have been characterized by the National Research Council for risk assessment. You do a hazard identification, and then you do a dose response assessment. And this is a part that I don't think genetic toxicologists have been doing very much, if at all. Well, I actually did it a couple of times, with George Douglas, for example, but not very often. But really important is this idea of exposure that gets brought in. You got to figure out who's being exposed, who's susceptible, what's happening to these people. And you bring all these elements together in a risk characterization. And that's what we're going to talk about at the end of the day in the discussion. What are the elements you're going to bring together in your risk characterization so then you can actually go to the management for them to make decisions. There's this policy, any type of actions that you might want to do to mitigate any type of exposures. This is the mutagenicity testing battery. And I think what's happened with this is that initially when we put this out in 1991, the focus was on heritable risk. This was a screening battery to see if there's any activity for mutagenic activity. But more importantly is that if there was, then there were follow-up assays to try to figure out was there going to be a problem in the germ cells and ultimately quantitate that activity. Well, now these two assays are not done much anymore, so that's sort of withered away. And we really don't pay too much attention, really, in the regulatory realm anymore. But I think we've got to re-emphasize that heritable risk is really important. And I think that's the message we're trying to do with this 50th anniversary, in a sense, really. And I think that we're going to be talking about that tomorrow morning. I think it's going to be a very important thing to resuscitate this idea. But mainly what's happened is that this battery at top sort of became the rigid way of doing mutagenicity testing. It was either yes or no. You have activity or not. Well, when I used to go to my bosses and everything, they say, well, we have mutagenic activity. They go, what are we going to do about it? And it was really like a big shrug. And we go, well, what we do is it'll support a cancer case. And if there happens to be no cancer data, we'll just go ahead and trigger a cancer assay. And that's what's happened, I think, over the years. And that's really where I think we are sort of stuck in this conundrum. We've got to break out of that. So, what we did is, under the auspices of the HESI Genetic Toxicology Committee, decided to try to do a clean sheet approach. It's not a clean sheet to clean you up or anything. It's really take a clean sheet of paper and say, okay, based on what we know now, how would we proceed in our thinking and try to figure out, is there a real problem or not? So the goal was to develop a genetic toxicology testing strategy from clean sheet incorporating new science and technology. We published it in this particular paper. It gets back to that initial battery I just showed you. There's really no longer a single approach in all the molecular mechanisms that can cause a problem. It's not just cancer anymore. It's, as Joel said, it could be neurobehavioral type of stuff. It could be other things like asthma and other outcomes. So we got to think about where the mutagenicity activity is relevant to the particular health outcomes. So what we want to do is become relevant to human risk assessment. This is the framework that we put together. This is in a cartoon fashion. But it takes into account a lot of the risk assessment elements that I showed you a little bit earlier. Starts out with the planning and scoping, including the anticipated exposure. This sort of give you a quick idea where you want to focus your particular testing and what is the problem you're trying to solve. So you ask a lot of risk management type questions like who's being affected, how much is out there, what's going on, and you want to try to figure out how you want to focus your approach. You just don't want to just do a testing battery, just say I'm done. What you want to do is then decide, you're building a knowledge base and things which you know, you build a biological argument for how you want to approach your testing. This is where AOPs and mode of action type of stuff is very helpful to give you it gives you an idea about key events that you might want to target for your testing that are relevant to the endpoint that you're looking at, in this particular case, heritable risk. When you do the assays, you review the results, and I'll get to this part later. This is actually starting to quantitate the information. For most of biology, they do this all the time. Even the genetic toxicologists do that. They kind of understand we got to do some testing. If we find some positive results, we kind of have an idea, well, we might follow up, try to get a mode of action question out of that. But we're going to do this now more in a more systematic way. I think the clean sheet part about it is to break the mold of just doing the exact same testing battery all the time. You got to figure out what is going to be the most appropriate test to do. And that's what we're going to be asking today, as you listen to all the talks today, is which one of the testing things that you've heard today are going to be the most relevant, you think, for decision making down the line. The other part is the real clean sheet is genetic toxicologists, as I said, really haven't quantitated much. And they don't do much dose response analysis. So now the tests not only have to be done, usually at high doses, it says, okay, yes or no. We want to do a dose response analysis where we can get some type of idea of where the dose response curve blends into the background, and we start doing some quantitative analysis. And I'll talk a little bit more about that. I'm going to go through the steps fairly quickly because I want to get to the endpoint later. The main thing during your planning scope is you're trying to construct risk management questions. Why you're testing, what regulations you're going to be addressing. This is really important. What exposures you need to take into account. And then this is more like planning. Who's going to do the work? How long it's going to take, how much it's going to cost, and that type of thing. So these are all things you think about up front. And while you're focusing in on, what is the actual problem, and then you start looking at what are the actual data that's out there already. Well, for tobacco-related products, we have a ton of data already. But we just kind of, in this particular case, we want to sift through it and try to figure out what's going on. But for new chemicals or exposure type thing, you start looking to see if there's other types of information. You go through all these tools, anything like PBPK modeling, QSAR information, silico assessment. You look at your mode of action information, see if there's any test results that are there in any existing human data, which is usually the ones most people like to have. I think you're going to hear a lot about that today as well, which is nice about the tobacco smoke as an example. And then, so this is really sort of where you want to try to break out of the rigid thinking of just doing a standard battery of testing. You want to build an argument for what specific testing strategy you propose for your specific compound. And you can look to the mode of action, AOPs, for guidance where you want to do the targeted testing. So it's a kind of, again, clean sheet, break away from the rigid way of testing and do that. And I think there's going to be a lot of talk today about what's tested and done. I'm not going to read any of these two slides. Mode of action and AOPs, I think most of you are familiar with that. An MOA characterizes a general understanding of how a chemical acts. So what you want to do is pick a particular MOA that is relevant to humans. It helps also identify susceptibilities, which I think is going to be very important today in terms of tobacco smoking, particularly if you're talking about children and the elderly and people who might be super sensitive to tobacco smoke. It also helps identify which tests you can do and ultimately help define how big of a safety margin you might want to put on your quantitative analysis. So, the AOP is a similar type of concept. It kind of gives you the same kinds of thinking and output that the MOA is in this particular case. So then you go ahead and select your assays and perform them. And again, you want assays that are pertinent to your proposed mode of action. It can be a few tests or several tests if it actually addresses the problem you are trying to address. In genetic toxicology testing, we do a lot of testing in vitro at first. But I think in terms of really trying to figure out real risks to you've got to finally start doing some in vivo testing. And I'm a little at odds with the current EPA approach of thinking. I've always thought that. And this is just a little thing I put on here just to remind me that there's a ton load of new technologies, some of these high throughput type tests. So there's, again, open your mind up to what are the tests that are available out there for you to use in trying to address the question. So after testing, this is where I want to start really getting into the discussion and help us with the talk at the end of the day. But you just review the results, and then this is where you start doing your dose response analysis. You're trying to determine what test results are used for determining a point of departure for risk management purposes. So we're going to use quantitative approaches, and the genetic toxicology community has pretty much started focusing on benchmark dosing approach for analyzing dose response curves. And I should throw a plug out. That would be a session that we're having on Sunday afternoon. It talks all about that. So if you're interested in that, I would really highly advise you to go to that. We have a really excellent slate of speakers. I'm not part of it. The guy who's going to tell us the real good stuff is Burke from the Netherlands, and he's like way out there in front of us. And this is important here, bringing the expected and actual human exposures. That's the other part of risk assessment. You don't have risk if you're not exposed to it. It could be as bad as anything, but if you're not exposed, there's no risk. And that's what the managers want to know. Are people really going to be, affected by this? So that's part of the risk assessment that we need to be well aware of, and I think tobacco smoke, and that is going to be a very good example because we know all about the types of exposure that are going on out there. You need to think about that today. This is just an example of a dose response curve analysis. I'm not going to go through this again. I'm on Sunday afternoon. But you do your dose response. You try to figure out a particular response that you really want to concentrate on that's kind of near your background. We call it a benchmark dose at this particular point at a 10% response above background, essentially. And there were two ways that we're essentially going to be using this point of departure, which is the benchmark dose. We do some uncertainty factors, or not uncertainty factors, I'm sorry. What you use is a lower confidence limit. You determine a lower confidence level of a 10% response. Then from that point of departure, you either use uncertainty factors, you create a reference dose, or then you compare that point of departure to environmental exposures of interest to calculate a margin of exposure. And so we're going to see if we can do some of that today, but I don't think it'll be too difficult to try to figure out which test. The thing you want to do is which tests you want to do to actually use the modeling procedures that we have in place. Here's some, just give you some references on how to do some of this. All right, now for this workshop, this framework essentially is to systematically organize and review the known methods and data used to study potential effects for tobacco smoke exposures. So what you're going to try to do is write down as you listen to the talks today, is write down the key information you think is going to be used to characterize and establish what heritable risks may be found. And then it will help you determine if there's future testing and regulatory policies if any are needed. Hopefully there are some in place, but if they're not, what would be some recommendations that you can use to say, yes, we can need to address heritable risk. Risk characterization. So this is sort of a little bit of a checklist of what you can go through today. The ultimate use of risk assessment is to provide risk managers and decision makers with facts and evidence that relate to human health. Okay? So that's what you need to pick out of the discussions today. So what are the key pieces of evidence you would present to support whether there's a heritable risk or not from tobacco smoke exposure? You just can't say it's bad. You know it's bad. You got to say, okay, what are the ones that are really going to be the pieces of, the studies, the data that show there is a true heritable risk problem. Then from those, what data sets would be the most appropriate for quantitative analysis? That might be a little more difficult because if you're talking about phenotypic changes and things, it might be hard to quantitate what that is. But we'll give it a try. And finally, what exposure scenarios from tobacco smoke exposure you should suggest are the most problematic? Here you might be asked to prioritize children first, identify susceptible populations later. So any relevant susceptibilities. Again, you might want to talk about what types of tobacco products you want to focus on. Vaping's becoming real big now. Is that one we really want to go after? Because that seems to be more of an acute problem now than the chronic cancer type studies. Or, long-term cancer, smoking. Are there opportunities to address the marketing of these types of products? And that goes into my next slide. I wrote an article for the Codex Alimentarius about what are some of the risk management options that risk managers kind of go through in terms of how to maybe address a particular problem. It goes back to the risk management question at the very beginning. What are the things they've got to address? Is it a certain population, or is it, a batch of chemicals? We got to get rid of some of the chemicals in the batch or something. So there are all sorts of options that they go through. Most people think about setting a regulatory level. Again, I talk about the RFD and the MOE. In the international world, they call it a maximum level. But you're not going to be able to set a maximum level a lot of times on an RFD in many cases. But there could be other things that managers could do. So let's just say some of the options we need to think about today. What could we do? Maybe push the regulators to do something, take some action. It may actually be a best practice guideline document, how to make a safer cigarette, if there's such a thing. Or a code of practice on how to manufacture things and things. Can issue advisory documents on safe intake levels. I don't know if there's a safe intake level for tobacco smoke, but you might want to determine, I'm almost done. You might want to determine, is 2 cigarettes too much a day? Is it 1? Or, there's a sort of a semi-quantitative analysis there. Require labeling. It's a big thing in tobacco on cigarette packs and things like that. Is there more use of this that we can do? Again, work with industry to reduce human exposure by setting targets. Make sure that there's only so much nicotine in a particular product or something like that. Again, you can invoke recalls, like get something off the market. These vaping problems, if things are a problem, you might just want to get them off the market. And the rest of these are just sort of education and training for people who might want to go out and advocate, for limiting cigarettes and smoking. We do laboratory research, but always make sure there's good communication among all the stakeholders and what's going to happen in the world of regulatory things. So, looking forward, so what today we want to do is making sure, are there any data gaps where follow-up studies could be useful? Or do we have enough information in all of this? Is there enough evidence already for any particular specific susceptible populations or life stages? And then finally, based on your knowledge of current regulatory approaches, do you think there's approaches in play that we can actually use right away? Or do we have to sort of petition or something, or make sure we can try to make sure that the regulatory policy is changed to emphasize germ cell risk? essentially follow up what Joe was saying in her beginning thing. We got to make sure that the regulators and the decision makers realize heritable risk is just as important as systematic risk. I think that's it. Thank you very much. Hope to have some good discussion this afternoon, so keep this in mind to organize your thoughts.
So we got 5 minutes. I'm going to try. Hang on. We got 5 minutes. So I'm going to try to moderate the time here to keep everyone on track because we have a lot of speakers. We don't want to get too far off track, but we're going to have 5 minutes. So
I think I finished.
Yeah, great.
So, as you're thinking ahead, how are you thinking of integrating the backwards with the exposome? We're not just exposed to the backwards, but it's first hand or second hand.
Well, that's
Risks associated with tobacco, what is the thought of a concept of the fatality?
well, absolutely. I think that's probably something that should be thought about today. I totally agree with you that you're not exposed to just one thing at one time. You're going outside, you're breathing all sorts of other types of pollutants, not just tobacco smoke. So how does that interact? Is there synergism or whatever? So the exposome is definitely a yeah. Yeah. Yeah.
If I may, I mean, Kerry knows this as a federal employee in several agencies, and you know this yourself. So even smoking in this country is divided between 2 agencies. So EPA has responsibility for just passive smoke, environmental smoke. FDA has responsibility for primary smoke. So already we have, I agree, we need to have this integrated assessment, and yet administratively and legally, so those 2 aspects of exposure are separated here in the federal government. Don't talk to each other about it.
Yeah. In addition, studies on complex mixtures. The EPA's been looking at complex mixtures forever, and it's like one of the biggest conundrums they've had. How do you put them together? Yes.
Yeah.
I think it would be, I think it's so ridiculous because it's so expensive. But the idea of establishing susceptibility period and using smoking as a model, considering the pure exposure. I think then one can assume that one can document that there are more susceptible periods that would relate to many exposure and that would lead to moving resources.
Yeah, because smoking. Well, smoking, makes you more susceptible to something else, or vice versa. That's a real problem. So, Abby, you might want to make that as a point to talk about.
Anything else? Yeah.
Okay.
So there is, as you know, I mean, there is data on exposure.
Well, there certainly is
Yes. But not, no, there are no clinical studies. But there is clear evidence of exposure based on, it coming off with radiation and all that.
Well, that's why we want to ask through today, do we have the studies about heritable risk? We all have them about lung damage and that type of thing. That's what this workshop's all about. Hopefully using the framework, you can kind of zero in on what are the important pieces to support that.
Thank you so much,
All right, thank you.